All right, welcome back.
Thank you.
Here we go again. So today we're going to get into a bit more of the foundational sort of concept things, but I think let's start with a quick recap in terms of in the last video, in the first video we talked about nanobubbles and their effects. In the last video we essentially made the claim, I suppose, that the glycocalyx ESL structure is this self-assembled series of interconnected tubes and almost sort of cell trafficking system, for want of a better term, that is, again, based on self-assembly. It's a dynamic system that allows for subtle through to very significant changes. And as a result of that structure, we believe that because of its connectivity and its ability to make all this sort of dynamic change, not only is it part of an expanded neural network, but it's essentially, the way I like to look at it is, I see the GC structure as almost like the definition of homeostasis, right? If you have this structure working within its normal range of parameters, and we can talk about what that is, then the way cells functions and communicate and interact with each other will be normal and health can be maintained. Is that a reasonable summary, do you think?
Yeah, okay, well it's a regulator, yeah. It's got the capacity to regulate because of its elasticity in changing permeability, flow, and so on.
Yep.
Yes.
But it's incredibly, although it's a structure, it's incredibly dynamic, and there's a whole range of things that can affect that change, and we'll start to look into that. But that's essentially the sort of claim, right?
Yeah,
okay.
It can change. It's a road system that can change the number of lanes and it can change the number of traffic lights depending on the chemicals that are going through.
Yeah, and so part of that claim then is it's almost infinitely more important than this sort of cell membrane, which we've spoken about, this 3 nanometer thick. I guess the general rule is extraordinary claims require extraordinary proof, right? So now I want to start walking through that. Well, how on earth do we believe all of this? What's it based on? And particularly looking at it from the angle of how much of this speculation, because at the end of the day, when you're talking about a dynamic biological system that only functions inside a living organism, how much of that are we speculating is true versus how much of it can we demonstrate are fundamentals? If that makes sense. So we've talked a little bit about some of those basic fundamentals, especially when we talked about nanobubbles and the effect of salt concentrations and amino acids, etc., and how they will affect the environment and how things behave in the environment. But today we're going to go through a basic crash course in self-assembly right through, and I think we will try to start with basic surfactant interplay and move through into the more complex world of polymers and ultimately look at the Nafion sort of example. So, okay. So, crash course in self-assembly. So let's start with really what we mean by self-assembly and the way we look at, particularly the way surfactants assemble and form structures. So we're starting here with this particular, and here we're looking at essentially a micelle. And so here we're looking at an example of a single chain surfactant. So a surfactant being a polymer that has a head group and a tail.
It's not a polymer. It's a
Sorry, yeah, so it's not a polymer. No, it's a single molecule.
It's a schizophrenic molecule that's got a head group that likes to be in water, it's hydrophilic, and a tail, like olive oil, that likes to be in oil. So it's got this schizophrenic nature to it. It wants to be between oil and water.
Yep, okay. And so because of that, whenever it's in a aqueous solution, right, environment, then the head group's going to be essentially attracted to the water molecules, and in this case, hydrocarbon tail is going to be looking to avoid water at every opportunity. And as a result of that, the forces involved in those hydrophobic, hydrophilic forces, and we talked about they're a result of the dissolved gas, but in all the environments we're talking about now, we are talking about environments that contain dissolved gas. So this concept of hydrophobic and hydrophilic is relevant, if you like, in terms of understanding the nature of the forces. The hydrophilic head is going to be searching for water molecules and the hydrophobic tails are going to be seeking to avoid them. That's essentially what's going on, yeah?
That's the simplest system. Surfactant is a mnemonic for surface active agent. So it includes soap molecules like these, but it also includes proteins and many other molecules that have got hydrophobic and hydrophilic moieties. So they're always going to be surface active agents depending on the
because
balance.
because that surface means there's a different forces applying on either side, being that hydrophobic, hydrophilic. So, okay, so we'll deal with the simple ones rather than getting into the more complicated multi-moiety kind of molecules. But so in this sense, we've just got essentially a binary. One likes water, one hates water. So because of that, the tails then, the way I like to look at it is the tails are trying to avoid the water because the water is otherwise unavoidable. So they're going to look to either bury themselves, and we know this from, it's like your basic detergent, that tail is going to sort of bury itself into the food and grab the food so it can hide from the water. Where there isn't an opportunity to sort of bury itself or avoid those, that's where these structures start to form, and in particular micelle, because essentially in a micelle all the tails are lining up together, yeah? So that they're, whether you picture it as the tails are being pushed away from the water or they're retracting to themselves or whatever, what we end up with is we end up with this sphere where all the tails are pointing in and all the head groups are happy in the water. And that is now balanced in the sense that those forces have balanced out to make that structure reasonably consistent without any other forces acting. Is that a fair way to think of it?
Yeah, except that it's very dynamic.
Yeah, well, in a biological system, it's bouncing around or whatever. So,
okay.
but if I did this in my sink and or in this glass and I just did that, then they would be stable in the sense that until there's other forces sort of acting on them.
Stability's relative. That's a thermodynamic concept, but it's very dynamic. It depends on the chain length, because they don't want to be in water, but they are in water if it's dilute. Entropy demands that. So as this slide says, that the lifetime of a surfactant molecule in an aggregate, it's in a small chain length like C10, CH2, 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 C10 surfactant, it's about 10 to minus 8 seconds. That's not very long.
So we are talking about extremely dynamic.
And a micelle itself is stable. They're constantly forming and reforming.
Yes.
The whole micelle only lasts about 10 to minus 5 seconds. So they're moving around and able to do things. If you go to a longer chain surfactant, like a double chain lipid, as long as olive oil, C18, it forms into a different aggregate, whether it's spherical, or cylindrical, or bilayer, and its lifetime in the cell membrane, which is made up of two layers of these things, is about 3 months. Right. So you're varying over an enormous magnitude. The cell bilayer, a molecule there lasts for 3 months in there, whereas a small surfactant molecule is buzzing around like a whole bunch of bees. But if you look at them, they're stable. They are stable at the same time. But you've got that overlay of a great deal of flexibility to exchange molecules from one aggregate to another.
Yeah, and so and again, I'm just sort of thinking of, without doing the math on this scale, but it's something like the lifetime of a fly through to sort of geological periods. Like we're talking that sot of scale, right? We're not talking about your age versus mine, or an elephant versus not quite that scale. We are talking about orders of magnitude different sort of thing. Yep. So okay. So all right. But I guess one of the important things then to understand leading from that is that because of these varying different constituents, and again we're looking at a single here. It's in this example, if all the chain lengths are C10, which in reality in a biological system there's going to be varying chain lengths of varying things and singles and doubles and all those sorts of things. But to keep it sort of simple, the other problem that occurs then is depending on chain length and single versus double chain length as well, is we get into this sort of concept of curvature and packing constraints. And so, and in particular the asymmetry of how these things sort of come together. So probably actually if we start with this particular diagram I know is one of your favourites because it sort of illustrates the asymmetry of the way these things come together. So do you just want to talk through around when it comes to bilayers, what's occurring here and what do we need to understand from a physical perspective is inevitable as a result of these structures?
Well, do you want to describe a bilayer first? Or
Yep.
in the slides, yeah. So you had a micelle, well, there's, depending on the chain length, how much volume there is to the surfactant and the head group. Depending on that balance, you'll go from a sphere to a cylinder, to a cylinder there, to a bilayer, and then multiple layers, and then even reverse layers in another slide down there. See, they're the reverse curvature. In that case, you have water on the inside and oil on the outside. So those are the simplest things you can deal with. But then when you look a little bit more closely, you come down to that other slide you had, which looks at a bilayer, and that has, it's very simple, but it has very profound implications, because if you take a bilayer and a cell membrane is made of lipids, it's got lipids and proteins, but the lipid component, that's the boundary of a cell, is a bilayer. It's two layers of double chain lipids that form a plane. But as soon as you bend that, you run into a problem.
Which obviously, sorry, just to make that clear, so this looks a little bit kind of two-dimensional and linear, but in reality, we're talking about a cell, not a perfect sphere necessarily, but spherical. It's a continuous layer, right? There's no end, start, stop scenario, because again, these tails are getting away from water, so that's why this sphere is being formed, and because of the sphere, you're going to get this asymmetry on the inside and outside,
The inner chains are squashed and the outer ones are stretched out for a given head group curvature. So the head group, the tails are repelling, the head groups are attractive and repelling. So this balance at the interface determines the curvature. So any curvature is going to be necessarily under stress because of that asymmetry, and that's really quite important. They're double chain surfactants, lipids, that form the cell membranes, but that's the situation, I think, to keep in mind.
Yeah, and so... Yeah, okay, and so there's going to be limitations then in terms of size and shape. So if we say, okay, well, depending on the chain length is going to determine how big this sphere can be before it falls apart, but also particularly, and I don't want to get too sort of caught up in the detail of this at the moment, but it's important to understand that, again, in a cell membrane, we're not talking about a single lipid, right? We're talking about multiple different types of lipids that have themselves different chain lengths and therefore different impacts of cholesterol forms a stiffer layer, and we'll talk about those things in a bit more detail as we get into the weeds, if you like, of those individual lipids. But I guess the important thing to understand here is all lipids are going to have their limitations because of their size and shape in terms of how much structure they'll allow, for want of a better term, yeah?
Yeah, so, and this is for the simplest case, a single lipid, single chain length, a single head group. So that imposes a constraint, which is really important for biology, in that if you form a vesicle, just a tiny balloon, it's going to be different inside and outside.
Yep, the inside layer of the balloon is different to the outside layer, even though conceptually we just see the layer of a balloon.
But it's more than that, because even with that simplest case conceptually, the inside of the vesicle, do you have a vesicle layer? Do we have one? I think no, you don't. Well, anyway, a single balloon, a vesicle that contains different things that react and so on, the inside is going to be completely different to the outside. There's an asymmetry.
Sorry, so again, I think just to clarify what I think you're saying here, so what's happening, so the environment, if you like, outside here, okay,
because of that
is different to what's in here. So the ions in there, the dissolved gas, the other lipids and proteins and everything else that's floating around out here is fundamentally different to what's in.
Yeah, so if you do a simple calculation, you can show that if you have a charged lipid, and many lipids are charged, then inside that little balloon, there's two things that happen. There's a minimal size to a vesicle, to a balloon, because the chains get squashed and they can't get any smaller. The limit to the size that they can take. And also, if they're charged, then if you do a little calculation for a typical 200 nanometer vesicle, then inside you can have a concentration of counterions to the charge on the lipid of 1 molar, but 10 to the minus 5 molar outside. So any container in biology, which is going to be hugely asymmetric, the physicochemical environment is different inside and outside. And the vesicle has a limited size. It can't squash down any further than that without turning into a different kind of structure.
Yeah, and we'll get to those in a second. So again, the analogy I'm sort of seeing in my head is almost a bit like, you can fill a balloon full of nitrogen at 1 atmosphere, and if you raise that balloon through the atmosphere, through the stratosphere, the forces fundamentally on the inside and outside are going to be different. The balloon's going to expand, and you're going to get a different effect as a result of that. You also then change the nature of that gas, or the starting temperature, and all those sorts of things. You 7 different balloons could have slightly different parameters that will all pop differently at different levels of the atmosphere as they sort of go into space. And again, that's a ridiculously simplistic view, but I guess making the point to say that if someone's holding a balloon here and says, if I let this go in and goes into space, without knowing all the details of what's inside the balloon, you can't really predict what's sort of going to happen, because there's so much difference of the inside versus the outside of those layers, as well as the fact that the balloons are made out of the same material and so on and so on.
Yeah, the geometry and the chemistry are intimately related.
Yes, but there is, at this scale, there is a physical limitation and a physical impact on these things that ultimately are going to determine what's possible structurally. Yeah. Okay. So again, I don't want to sort of dwell too much on the point of all this sort of complexity, but other than the point to say it really doesn't matter, single chain, the nature of the chain, dual lane, all these things do is affect the nature of the things that are possible, but the concepts are the same. We're still going to get that degree of asymmetry, we're still going to get these packing constraints, we're still going to get these different conditions either side of the layer. Okay, so again, a really important concept is curvature and the packing constraints related because of that curvature are really important aspects when it comes to looking at things like what's driving this dynamic structure that we're talking about. That's really the sort of point of this. Okay, so then if we look at then how does that form into structure, right? And we expand that out and say, okay, well, if this lipid bilayer has these constraints, it can only bend so far. When we reach limits of those things, what are the natural byproducts when these space limitations occur? Do you want to just sort of talk through some of the examples of what happens when we run out of room, for example, and those sorts of things?
Yeah. If they assemble into a vesicle, and you think of it as a balloon, like that, that's a section there, they will assemble naturally into 1 layer and 2 layers, 3 layers, so you get a multiple layered system, which is called a liposome. Lipids do that. So, but eventually, if you look at the different layers of these things, you go back to that original slide with a stressed layer. Eventually, if you imagine the layers getting smaller and smaller, when you get down to about 200 nanometers, typically, for typical lipids, you reach the point where the chains are going to break. They can't pack anymore. So they can ease that tension by curving.
So just to sort of clarify, I think what you're referring to is sort of this sort of concept of these sort of multi-layers, right?
Multiple layers.
So here we've got each of these yellow lines are a bilayer like this, and as we get smaller and smaller and smaller, we're going to run into that problem more rapidly, if that makes sense. Yeah. Yep.
So then it can ease that tension by curving both ways. You have a curvature one way and a curvature the other way. So the net curvature, the average curvature, is 0. So it's got the same curvature, the lipids, as they have in a bilayer, but they've got different geometric curvature, product of curvature as you go one way or another. So you have this, what's called monkey saddle. Yep. And so you can put these monkey saddles together that are now happy in their curved state. They've got the same curvature, average curvature.
So just to clarify on that too, and the reason why that's important is because, again, the forces are trying to balance, right? So if it's getting squeezed too much, something's got to give, and the give is, well, if I throw out and bend in these different ways, I can distribute that force, if you like, a bit more.
It's just a consequence of geometry and the fact that molecules have size and shape. So then that can form into much more interesting structures that are shown there in that slide.
And so as a result of this sort of Pringle structure here, which again, you know, from a forces and an energetic sort of perspective, that flip, if you like, from a curved to a Pringle in this case, is it right to think of that as a, not that everything's fine, but it looks the same right up until that point, and then it's now a completely different structure.
To a different structure. Yep. That's called a phase transition, a mini phase transition. Yep. And the structure, which is what's called a cubic phase, because they're cubic symmetry, you can see in the next slide if you like. And that's again pretty important. If you go down further than that, there's a picture of a cubic phase formed of phospholipids. You can see that everywhere you've got 0 curvature, but everywhere you've got curvature and you've got interpenetrating channels. And you can imagine that thing surrounded by a bilayer from which it originated, or a multiple bilayer. So you've got a thing now which is like a balloon, but it's filled up with a structure that it's got varying connectivity and channels going through it. So that can take...
So this could be an aqueous channel, this could be a sort of, lipid hydrophobic channel. Yeah.
There's multiple channels. So you set up an infrastructure which is much more intricate.
And I think it's really important to appreciate, this looks a little bit like, almost to me, like Lego and solids, but we've got to keep in mind, all of this we're talking about fluid, right? So this is one form of fluid versus a different form of fluid, be it gas or water or...
This same reasoning applies to electrons in metals and to solids.
I see, at this scale.
Yeah,
at this scale.
At the molecular scale.
Yeah,
but at all scales, actually.
Okay.
Because if you look at a termite's nest, it's bicontinuous.
Yes.
And if you look at a termite, we've had a slide somewhere there, a termite's nest can go up to 3 metres, but it's full of this bicontinuous traffic.
The termites build the nest based on this geometry.
Yeah, and they have a whole city that they drive through these holes. So in biology, you've got molecules doing the same as the termites.
And I've always found that really fascinating, because I know you've used that as an example to many times to sort of give an appreciation of scale. And on the surface of that example, it can seem like the termites have gone to the ultimate engineering school to kind of work out what's the best way to build this structure. But in reality, if you think about the way the termite builds the nest, he's just going to follow the path of least resistance, right? This is the way I sort of look at it as well. And so this is kind of almost like, would it be fair to say that these things occur more because of that kind of path of least resistance scenario? That's why we see them across all these scales? Why do we get the same phenomena at the termite level, who doesn't have a degree in engineering, let alone non-Euclidean geometry?
Yeah.
How does he end up with the same structure as this?
Well, he's probably as smart as the average biologist.
But is it like? Do you think is it likely that the termites just, again, following the path of least? He's trying to follow the conservation of energy.
They've got a lot of work to do to go carry wood back in, fuel back in, the same as the biological organelle. It's got to have stuff in to do things and stuff.
But he's not sitting, I guess he's not sitting down with his CAD program and working out how to do it. He's just pushing a hole in the ground, essentially, yeah. And so would it, to me, that it seems to me like it's like, well, he would push the easiest hole in the ground, wouldn't he? And the aggregate of that turns out to be this sort of structure.
Yeah, well, the inside and the out, I don't know how to put it, the physicochemical environment outside and inside, they drive each other. One's the source and one's the sink, so the sink and the source go together. And in this case, it's affected by the shape and size of the lipid molecule. They've got
essentially containerising this. They've got no other option.
They can't pack any other way.
Yeah,
but it turns out to be optimal.
Well, you couldn't pack any other way without introducing additional energy and forces to stop it.
You could introduce a different molecule of a different shape or a different lipid. That will change the microstructure.
Yep. So again, let's pretend this is sort of sitting in my sink, and this is balanced out now in that. Appreciating that all equilibriums are temporary, right? So. But in reality, if I go and then drop even a single sodium ion or a single change to this environment, it's going to have an effect.
Well, it would, but you need to have a chloride ion to go with that.
Yeah, okay, right. Okay. So again, the important thing about this picture is to appreciate that what we're talking about here isn't solids and air or solids and water per se. We're talking about what essentially are going to be dynamic channels of aggregate fluids, in this case, that are either hydrophobic, hydrophilic, predominantly in this sort of structure, yeah?
Yeah, and they're universal because you've got to, and they can transform from one form to another very easily. There's a whole literature about this, about the bicontinuous geometries that are in fact the language of nature. Yes. We saw in one of those earlier slides where you had spheres and cylinders. And that's what the language that people use, the Euclidean geometries, spheres, cylinders, so on. But they're too simple, and nature, they don't have much variability with them, but nature exploits these bicontinuous structures to set up much more interesting infrastructures that it needs. Yep.
So this sort of non-Euclidean geometry and sort of the way more complicated geometry is what nature harnesses, essentially. Yeah. But they're the result of a lot of so Euclidean geometry is much more about like unitized symmetry, I suppose, and the same sort of units and measures all the time. Whereas nature's, because of that variability, has to build, much more interesting, I suppose, or less. Mathematically balanced, if you like.
It does it automatically
because of the different forces and stuff involved. Because it's not all one chain length, one size. Yep. And the environments themselves are dynamic. Okay, so again, when we look at sort of the effect of this, so we're talking both within, in this case, a cell potentially, because that outer bilayer, sort of sets the limitation, if you like, in terms of total internal geometry, and therefore within that cell, depending on what's already in there, is we can, and that cell membrane getting bigger or smaller potentially, is you get this sort of change in what's the structures, if you like, that are internal to the cell, and those, because of those these other ones we looked at, sorry, the multi-layer scenario. Whenever you get this sort of these bilayers packing down to smaller and smaller levels, you're going to get these phase changes. Yeah. And those phase changes, am I right in saying that the point of flipping of these phase changes is itself it's a little bit more like the knife's edge balance, as opposed to, it doesn't require a huge shunt, it's just a small change in conditions once those conditions have lined up, yeah? Well,
here, what you can see, originally, if you think about the origins of life, you'd feel multiple layers of bilayers adjoining to multiple layers of polymers. And then if you look at that thing and it splits down into, as soon as you curve it, it turns into different kinds of organelles that are multiple layers, but the organelles can't pack when the radius gets too small, as we've been through. Yeah. And so then the inside of that thing that vesicle has got a structure which is bicontinuous and arranged to do all kinds of jobs. So you can see that a multiple layer originally in nature breaks off and to form all these different organelles surrounded by several layers of lipids.
Yep.
And polymers.
And so a cell had to
the
essentially
cell-
self-assemble because of these forces.
The cell self-assembled because it had to, because of geometry.
Yeah.
That's the first principle of chemical engineering. Volume. You have to conserve volume, and you've got to conserve the interface between two different regions.
Yes.
And so that balance is automatic, and it's automatically done.
Yep. But it's also done in a way, I know you used the origami example, particularly when we looked at some things like lung surfactant, etc. It's done in a way that requires the least amount of, almost like the least amount of force. Things fold and form the shapes that are the easiest to form. Would that be the way, yeah? And so these structures, the reason why they're so ubiquitous is because they're the easiest to form when you look at those relative sort of packing.
Yeah, it's like the Chatelier's principle to things will follow the path of least resistance. Yes. But in this case, it's decorated because you throw in a different structure to the molecules, different sizes.
Yes, but so just to explain that principle for those playing at home, is like that's also a principle that basically says, regardless of how complicated a system is, if it can find a new sort of equilibrium, new sort of balance, it will basically.
Follows the path of least resistance.
So going back to my, I'll throw a sodium chloride ions in, throw some sodium chloride into that scenario, it will perturb the system, but it will only perturb the system until it finds a new structure it's not going to send it cascading onto the, and blow up the whole universe because I dropped an ion in the wrong place. It's just going to find a new structure and that can satisfy these new conditions.
Yeah, if you like. Well, it could be catastrophic if it's right on the edge.
If it's right on the edge of everything, that's on the edge of everything.
Well, the structure of a rod and cone in the eye, they're made of multiple layers of lipids. Sure.
But it would only kill you, right? No, it's not going to wipe out humanity
it changes from one form to another. Yes. It's on the edge, poised as a multiple bilayer, the rod and cone. Yeah. Along comes the photon, and it hits something called rhodopsin, which is poised to receive it. That changes its structure, and it opens a gate, and it's a cascade. The whole multiple layer switches to a cubic phase that's got open passages that allow sodium to pass through and go through to the nerve. And then after that's done, you get this cascade that switches back.
Okay, so we were going to get to that, but we may as well, since you've opened that particular box, so again, I look at that as a whole series of almost gates down the corridor sort of thing, that when one door opens, it naturally opens the next one and it flows along easily and flows back, because each of those doors, they might weigh a ton each, but they're so finely balanced that all it requires just flows back easily.
And that occurs universally in nature. And
yes.
The geometry that changes that is by something called, in topology, or something called the Bonnet transformation. Yes. But it allows things to flip from one phase, a closed parallel lipid, to an open phase where you've got channels in it with extravagant ease. And that happens very easily. It can flip one form to another, back to another. Yep. And it's a universal principle that happens with electrons in metals, superconductivity all over. So what's missing in the general thinking in biology is awareness of these bicontinuous structures, hyperbolic geometries they're called.
Yeah, okay. And so just to sort of round out that analogy, because that's forming a good image in my mind of the relative simplicity. And keep in mind, I suppose, with all of these things, we are looking at survivor bias, right? We're looking at this version of things because it's the one that worked. And we're not looking at all of the layers that stacked upon each other and didn't form things that worked and, collapsed into different things. So again, I'm looking at this sort of, because we use that example of the rods and cones sort of example from vision. So you have picturing a corridor with these enormous doors, right, that might weigh many tons, that like the Egyptians sort of worked out way back in the day, if you put enough counterweight on one side, you don't have to push very hard and you can move a big object. You can swing freely and backwardly just sort of by by relatively small addition of force, right?
I was going to question the exceptions about that.
Well, even moving the big steel gates in the castle, right? They put enough counterweight there. The amount of weight to move the drawbridge up and down is relatively minor now because it's, you know, so sort of relatively finely balanced. But so because of that, if there's a small change at one end, it can open up all those channels. I guess my point here is a whole heap of things can flip or occur energetically with almost no effort, and can flip back with almost no effort because they're already so finely balanced. But if you knock one of those doors off its hinges because you've introduced, and we'll get into that when we look at disruptions, etc., if you disrupt that balance at some point, that's going to then stop that or interfere progressively with that, in this case, signal.
Yeah, maybe a better analogy might be the ocean. If you think of the ocean as the interior of a cell, and you think of the beach as the cell membrane, and you think of the outside as the superstructure that lines the cell too. If you think of just the tide coming in, you're opening up the cell, the sand, you get different flow through. Sometimes you get a lot of flow, and depending on the plants that are occupying the other side, which is another structure, you've got this continuity and interchange.
So your beach grass being your sort of glycocalyx or your rock head or whatever sort of things. Because I guess in this analogy you're sort of looking at the, I was looking at something that's a bit more specific and containerized, but you're talking about something that itself is a dynamic, moving thing that's sort of ebbing and flowing. And as long as there's enough, grass holding the beach together and the wind hasn't picked up too much, then the beach is going to be okay. But if you get that massive thunderstorm or that king tide or tsunami sort of thing, then you're in all sorts of trouble, regardless of how much grass you've got on the beach or what rock wall you've put in place. Yeah. Okay. All right. So I think hopefully now we've established this idea of why these structures or how these structures form, and then running that out the complexity, I suppose, of if we then take from a relatively simple example of a single or double chain lipid with a relatively small chain through to polymers. You made the point earlier that polymers are essentially conceptually the same sort of thing, except we're talking now much larger molecules with different types of moieties as well, so different sort of head group effects. So we might delve into that.
One thing I'd say that we've made this discussion very, very simple. Simple in the sense that we haven't been particular at all about naming any particular molecule.
Yes.
Like there are many lipids, double chain surfactants, that have got names, phosphatidylcholine and phosphatidylethanolamine, and molecules like sodium and potassium and lithium, and oils like unsaturated oils with a double bond or saturated ones. So all of these molecules have got names, and the first stage of science is to name things and then to recognise.
Or categorise them so that you can understand what you're talking about.
So I mean, you have all these molecules of different chain length oil, but then you recognise that they're all oils. They all have the idea that they're hydrophobic, they don't like to be in water. So this kind of theory you've got to steer a middle road between, there's a high road of mathematical physics, something called statistical mechanics, that formalises all this and produces in the end something called thermodynamics. So it's a generalised theory. But it's too complicated for anyone to understand. Everything's there in the mathematics and the mathematical physics, but you lose the you lose the trees for the wood. But that's the opposite to the other way. We're trying to make it simple. We don't want to get stuck in a simple way. We want to keep the woods and keep the general overview right. But we can make that too simple because what we've been going through, all the molecules are different. They've got different chain length. C16, C18, or CH2, 16, CH2, 18, so or different head groups, sodium, sulfate, whatever. They are all different, and because they're all different, slightly different, they'll pack different ways. So because of that slight variation, there's an enormous diversity to the structures that can form. All right, so they can be, we've eliminated the general kind of structure that can form,
Yep.
but the diversity that we're able to use, like if we get a drug to perturb the system, is as diverse as the edge of the ocean with all of these things. So keep that in mind. We're trying to keep an open structure, but you can see the whole structure emerging. And it's simple overview, it's beautifully simple, it's all organised, but at the same time we've got enormous diversity that once we've got the structure set up, the basic structure, we can employ and use it. And we can change the coastline if you like.
Yeah. Okay, look, that's a really important point because, again, in my mind, what we're trying to do here is exactly as you outlined at the beginning, you know, not close the gap per se, but explain how those extremes come together to meet in the middle. And you've often talked about this with me around, mathematics being the original language, particularly of, chemistry and sort of biology. And one of the problems with mathematics is it's a tool with a practical use that if you remove the practical use, then you can go off and do some really wonderful, interesting sort of thought experiments. But at the end of the day, you need to understand which concepts of that are useful in terms of trying to understand the physical world and which ones aren't, which are just sort of thought experiments for the sake of it. And to the other end of the scale, categorization is useful because if we don't understand it, if we can't chunk information down, we can't build our knowledge and assemble into complex scenario. But regardless of how far we go down any particular reductionist rabbit hole, we always need to sort of peel back and say, at the end of the day, what is it holistically we're talking about here? We are talking about cell membranes and cell membranes in this sort of, with these environments, etc. And the fact that we might be talking about the way molecules interact, but those molecules are surrounded by billions or trillions of other things that all have those different interactions. And therefore the degree of complexity is, as diverse as the universe itself. And so we need to keep that in mind as we sort of wade through. Is that sort of what you're saying?
You've got to extract from all the mess that we've got, all the different kind of molecules. You've got to abstract certain principles that...
Yeah, so we know that these things behave in these kind of ways and they'll aggregate in this sort of way. What we're not saying, and we won't say at any point in time, therefore, we can know with a degree of precision, first of all, what's even there, let alone what those all interactions will be specifically. And therefore, the result of this is we've come up with this miracle silver bullet that will restore every balance in every kind of way. That's just not how it works, right?
No, well, all of these organising principles come from the fact of hydrophobic, hydrophilic, yin and yang.
Yes, and walking the line, as they say. Yes, yep.
So it's the balance between those two things that gives you the flexibility to have all these organising principles, which is just size and shape, packing, as well as the hydrophilic, hydrophilic balance at the surface. That's all that's needed. And people lose sight of that and get completely lost because they don't have any organising principle.
And they don't tend to look at it from that holistic point of view of saying, here I've found some sort of biomarker or some sort of measurement even to the degree that that's the degree of accuracy. Let's pretend in this particular example that's the case. Even if what you are measuring really is what you think it is, really has the effect that you have, and a range is not right or normal, the idea that changing that in itself will be the answer to why things are out of balance when you don't understand the underlying structure. And I guess this is the central point of what we're trying to get across in this series is if the underlying structure isn't behaving at least in a degree of normality, then changing one parameter or something around the edge is going to have an effect, absolutely. And we'll talk about many of those examples as we get sort of further into this. But if the road network's not working optimally, adding a special car isn't going to help. It might get you from A to B once, okay, but it's your road network's still cactus, basically. Yeah. Okay, so let's continue on with this now. I think let's move a bit further up the complexity hierarchy and start to look at now polymers, or more complicated molecules, I guess is the way it's probably easiest to start thinking of this, where we're going from simple binary, head group tail scenario to multiple versions of this. Because really, at the end of the day, what we're talking about with the glycocalyx is a series of polymers that have certain, properties, if you like. And because of those properties, those polymers can now behave in a much more sort of dynamic three-dimensional way, in the way that they sort of aggregate and cooperate almost, sort of thing, right? So,
maybe you should tell people what the glycocalyx is.
Well, I'm assuming that if they haven't watched those the early videos to explain their structure, then that's the prerequisite to this, right? So we are talking about it's specifically, I think, because I want to move on to the example of Nafion and why we think Nafion is such a good analogy for the glycocalyx and how that kind of works, right? And then the work particularly that Nikolai's done around explaining the different dynamic activity and sort of size, shape, into exclusions and that sort of stuff of Nafion, particularly when we look at, deuterium and salt concentrations, those sort of things. So if we just do a quick recap in that, we're talking about, the difference between, say, Nafion versus the polymers in the glycocalyx. So we're talking about, in the glycocalyx, we're talking about, polysaccharides, so, hydrocarbon sort of change with these sulfonated moieties sort of a lot long, giving it the ability to form those helices we talked about in that earlier video, right? So, whereas with Nafion, let's walk people through what the structure of Nafion that's relevant to understanding why that's analogous to the glycocalyx.
Okay. Well, you want me to?
Oh, you to, yeah. So, well, I mean, I can start if you like. So again, we've got a fluorocarbon backbone, if you like, in sort of Nafion. But again, we have these sort of sulfonated moieties that are part of the structure of the Nafion.
Okay. Well, let me say this. The glycocalyx is the complex structure we talked about. It's made out of sulfonated polymers. Heparin sulfate is very common, occurs all over the place in medicine. Another one called chondroitin sulfate and several others. And the polymers are just like a rope with sulfate groups along them. And Nafion was a polymer invented 50 years ago. And it's the polymer that's used to make fuel cells for hydrogen. And it's made out of Teflon, a very hydrophobic fluorinated carbon with fluorine instead of carbon and hydrogen. So it's fluorocarbons attached to which, along the rope, are lots of sulfonated molecules. And it has some extraordinary properties that were not remotely understood till very recently. And they led to an idea that to explain their peculiar properties, you had to have a different phase of water, a new phase of water. Mm-hmm. So this was developed by a guy called Jerry Pollock. And he invented that for reasons, because he couldn't understand it any other way, its peculiar properties. And it turns out to be a mimic, if you like, an expanded version of the glycocalyx and the endothelial surface layer of cells. It has a so-called exclusion zone from the bulk region, which stretches out for hundreds and hundreds of microns. And it's got these long, thin cylinders that defy any kind of imagination. And so what the hell's going on? And inside the bulk medium, it's got, again, connected cylinders that are made up of winding up of the polymer with the sulfate on the inside and the Teflon on the outside. So you've got this wormhole thing with Swiss cheese through which ions can go. And because they're all sulfate and multiply charged, high degree of charge inside, you get hydrogen ion and hydroxide ion able to pass along through this this structure very easily. So that's connected to this giant exclusion zone, which is fairly sparse and expels colloidal particles, red cells, T cells, and big colloidal particles. So the question is why it does that. It doesn't fit into any otherwise known theory of self-assembly.
So let me just sort of just so we're clear in my mind, we're talking about so here we've got our chondroitin sulfate, heparin sulfate, GC, that helix structure we talked about in the previous video, creating these channels in the glycocalyx, and this exclusion zone which we showed in detail in terms of like keeping the red blood cells and the bulk medium outside.
That's the endothelial surface layer for cells, which is the same way. It's been known for many, many years. It's just a fuzzy layer on top of cells. Nobody's known its structure or the structure of the glycocalyx. The glycocalyx is about 50 nanometers for T cells. The endothelial surface layer is a micron, it's 20 times bigger. And the cell membrane, we keep saying this, is only 3 nanometers, it's tiny. So Nafion's the same, it's an artificial polymer.
Sorry, just to round out the comparison, so Nafion again, big long rope, okay, this time with fluorocarbon, so even more hydrophobic, okay, and it has these same, hydrophilic orientated head groups, so it forms a similar helix. The difference in this particular case is we're talking something like 300 times the scale, right? So we're talking about a much larger molecule, but the same properties. And you get this same effect if you get this channeling, but you also get this exclusion zone effect of the relation of these reverse helix scenarios similar to what we're seeing in the GC ESL kind of.
Yeah, well, we can deduce the nature of the glycocalyx and the endothelial surface layer from looking at Nafion, because it's the same kind of structure, but written on such a large scale that you can do laser scattering.
You can actually see it in the sense that we can measure and and that's in particular the work that Nikolai has done, particularly in that Nafion paper, which we'll provide a link to. But again, I want to sort of look at exploring. Okay, so we have these channels forming in the bulk medium, and we have this sort of exclusion zone with these reverse helices, as we sort of describe it. So, and that again is the same thing we talked about in the glycocalyx, in the initial glycocalyx video. That's a result of these moieties and their hydrophilic properties versus the hydrophobic properties of the backbones, creating these structures based on the self-assembly sort of principles we were just talking about sort of early, but now in a much larger scale with a more complex polymer. But the effect is the same because we still have that hydrophobic, hydrophilic balance. Yeah?
Yeah, the bulk polymer without water dry is just like a tangled mess.
Yes.
Like Teflon.
Yep.
It repels water. But when it's filled up with, put in contact with water, you've got water and you've got the solid polymer, but the polymer unwinds and forms these threads, and it coils around to form coils of polymer with the sulfates on the outside and the Teflon on the inside. So it's got a hydrophilic stretched out thing. It's like teasing a thread out of a wool jumper like this. You pull out these threads. So they're stretching out into the water a long way, and inside the thing swells and produces these coiled connected tubes inside through which ions can travel. And those coils can expand, get bigger, or connect.
So radially expand.
And connect more or less, depending on the nature of the ions, and how they interact with the sulfate of the group of the helices. So that's the picture of how it self-assembles, and it's really very interesting that the threads stick out for a very long way, and that doesn't make any sense without understanding that there's different kinds of collective forces going on that people haven't known about before. I was going to say too, for the Teflon case, with the Nafion case, it's really interesting because the threads stretching out, they wind around and they form clumps like ribosomes of DNA. DNA it's got winds up. The DNA compacts into the nucleus, and you've got a strand of DNA in the nucleus is a metre long. So how the hell this thing goes from a metre to something yay big? It's just it's the same kind of thing. They recognise each other. They interact with each other with very, very strong forces that people haven't recognised. And they form these ribosome-like things too, that they can coil around. Yes. And the coils coil around the coils, and the coils like a telephone cord.
So it's one big long thing, but it comes...
But so this study of Nafion is important for fuel cell technology, if you want to do it. But people haven't bothered. Well, they've tried to, but they haven't got very far with it until just recently. And the complexity is such that, as I said before, that Jerry Pollock invented this fourth phase of water, which is like another celebrated thing called polywater, which stirred a lot of trouble. But there isn't any fourth phase of water. There's just something even more peculiar, which we've identified due to different forces. And those same forces are operating in the biological scene, in the endothelial surface layer. So it's completely new forces that have come to play that are many body interacting forces that lie at the surface of the cell. So the Nafion gives us an insight into that.
So I want to sort of go into the weeds a little bit on that, because I think this is really important to understand, again, not so much the detail what have, but the nature of how the changes of environment, and particularly we want to talk about things like salts, and deuterium as some very strong examples of how and why changing in those background environments change the effect of from the chirality to the swelling, if you like, and the effect of that on the exclusion zone. So maybe if we start with, if we look at, and maybe this, starting with this table, what we found here is changing salt-specific ion effects, essentially, is we get a different change to the way the Nafion coils, in this case, are behaving, which then has an effect on the exclusion zone. Do you want to kind of walk through, sodium versus potassium maybe as an example, or why these specific ion effects and how they affect this, what ultimately we see as the exclusion zone?
These are demonstrations of something that's called Hofmeister effects, the universal everywhere in chemistry and biology, or specific ion effects. And it's a bit of a mess for the last 150 years, because all of chemistry, physical chemistry, started with the notion of just electrical forces, Coulomb force. Positive and negative charges interact with electrostatic forces. And so they made all these theories which still lie at the basis of all the physical chemistry. Notions we've got of pH, of buffers, of electrophoresis, of zeta potentials, all these different concepts that we use are based on and draw on an intuition that derives from electrostatic forces alone. The first 7 Nobel Prizes were given for work on electrostatic forces, solutions of electrolytes. So it all developed from there, and that lies very deeply in the heart of physical chemistry, and it's gone over into biology too. So when these theories don't work, and they don't work beyond very low concentrations of salt, you call them specific ion effects, which is a mnemonic for saying you don't know what the hell is going on.
But they are specific to each ion, so you get just as long as each one has its own rules.
And they are specific ion effects because the theory, as it developed, ignored quantum mechanics, and quantum mechanics gives rise an explanation of why the atoms are all different and why the ions are different.
Yeah, and we talked a bit about that, obviously, again, when we talked about the nanobubble section, and that it's not just a charge and size and shape, it's these.
So with the polymer, it gives them. Well, many of these effects are huge. They are for microemulsions. That's oil, surfactant, charged oil, surfactant, water systems. But if you change the counter-ion, dramatically the whole thing changes, the whole structure changes. And similarly with Nafion, it's another example of how these forces are highly specific and depend, in this case, because the sulfate is the operative anion, the Teflon sulfate is the polymer, the Nafion. So you've got all these sulfate things along the rope. But so if you change the positive charge that neutralises it, hydrogen, lithium, sodium and so on, then the interactions of those different positive charge cation with the sulfate change the structure of the polymer, how it winds up into cylinders or unwinds. And the differences are very large and quite dramatic. And that's useful to see because in biological cells, the difference between the inside and the outside is very different. Potassium is 98% inside a cell, sodium is 98% outside a cell. And we have this very peculiar difference.
So this actually might be a good time to do a really sort of simple walk through these partial phase diagrams because, again, they look really complicated, but they're relatively easy to understand, at least conceptually, I think, if we walk through it properly. But
no, they're not easy. It took me a long time to understand them. So
yeah, but once you explain them, they're relatively easy for a lay person to understand conceptually what's going on. I'm not professing to understand it at the micro level, but how about we walk through this in terms of my understanding of what this is trying to get across to the level of detail. So we're talking about, well, let's look at hexane as an example. So
we're talking about a mixture of oil, water and surfactant.
Yep. So in this case, this is 0 through to 100% concentration, 0 through to 100 and 0 through to 100. So this is inside here at any particular point, we're looking at the relative percentages on these axes. And my very simplistic sort of high school science version of this is as these concentrations change on these, in this case, the different concentration levels, is you get different behaviours as a result of the fact of the changing interaction of forces, etc. And I think the important thing to understand out of this is that when you reach thresholds, and maybe I'll get you to talk about what some of these thresholds are essentially, you get, in some cases, drastic changes in the resultant, say, force properties or outcomes or the way that they'll interact with their environment, be it from a viscosity or conductivity sort of perspective. Is that at least a reasonable sort of summary of what they explain?
Well, they're capturing what kind of structures form when you mix, in this case, just the three components, oil and a surfactant and water. Mm-hmm. In this case, it's a double chain surfactant, cationic, positively charged. And they'll assemble into different structures and they can form one phase, a single clear phase called a microemulsion, or they can assemble into a microemulsion and an oil and a water phase. So they can create 1, 2, 3 phases. So wherever you are in the phase diagram, anywhere in that triangle, that's a measure of how much you've got, surfactant, water or oil. And depending on the ratio, you get these different structures. The curved regions are a region where it forms a single phase. You take oil and water and surfactant, it looks like a cloudy mix in a test tube. Mm-hmm. You just add a bit more of something else and bingo, it'll turn into a single clear phase, which that pinky area all the way around is describing.
Yep. And so that's almost like the threshold we were talking about before. That's the sort of knife edge where everything looks the same, and then bang, suddenly we've got it completely.
Yeah. Like it'll flip from 2 phases to a single phase. Yep. Like gas and liquid. Yep. But what's interesting about them is that depending on the chain length, depending on temperature and so on, the microemulsion region, which is single phase, has an enormous variation in physical properties in its nanostructure. And the difference in the physical properties are such that you get changes in conductivity as you vary across that line.
Across this green line here?
Yeah, well, the parallel, the other line perpendicular to it, the one crossing it, yeah. Sorry. As you vary there, you're changing the amount of oil and surfactant. As you measure on that line, that's a constant. You're varying the oil content at a constant surfactant water level. Yep. And so when you do that, the conductivity varies by a factor of 10 to the 11th. That's huge. 10 to the 10th is huge, less huge, but 10 to the 6th is a million. Yes. So 10 to the 10th is 10,000 million. Yeah, so 10. So 10 to the 11th is 10 billion. Yes. Well, so that's a lot. So that's the changes, the structure, the conductivity. The conduct it's contributed because the clear liquid is made up of conducting tubes of water. As we talked about with those biocontinuous. Just like that surfactant, just like the Nafion that we saw. Yeah. And so it changes, the conductivity measures how connected those tubes are and how high they are. So it changes by that enormous factor. And the viscosity changes by a factor of 10 to the 5th, that's 10,000, 100,000 as well, across a single line. Yes. So everywhere you're changing just by the mixture and how they pack, you're changing the microstructure, the nanostructure of those. So the difference between, say me and Elon Musk in this example, right, in terms of net balance sheet, is a small change in those parameters, and I could flick and suddenly I'm Elon Musk's balance sheet. So you're changing the curvature because you're changing the length of the oil. Hexane has got 6 hydrocarbons, octane's got 8, and so on. Decane's got 10. So depending on the length of the oil, it changes. Depending on the surfactant you pack, you're packing, and then the packing determines the nanostructure, and that's a mimic of what happens in biology too, in the glycocalyx and inside the cell as well. Yeah. And so the I guess, and again, the point here, I suppose, is almost irrelevant changes, subtle changes at the right relative concentrations in this particular case, is literally that change. It takes an average person and turns them into Elon Musk's balance sheet as a result of what is a... much bigger than that. So much bigger change. And so the same thing happens too as you change the salt. If you add salt to the system, or you change, in this case, the surfactant has a counter-ion, which is bromide. Yes. It's got what's called a quaternary ammonium head group. Like sodium, potassium and so on. Quaternary ammonium is just like that, it's a cation. And the counter-ion to it is bromide, like sodium, bromide, sodium chloride. So if you change the surfactant from bromide to chloride to whatever, the whole structure changes again. Tiny differences in the counter-ion, sodium, potassium, so on, have a dramatic effect on the nanostructure that you make. But the whole principle stay the same. Yes. The state of self-assembly is the same. They're all encapsulated in something like that. And going through the whole system of self-assembly, you've got the universal set of principles, you've got surface forces opposing each other to give a curvature and bulk packing constraints. And that's enough to determine the whole thing. Yep. And as a result of those limitation constraints, when you reach the edge of those limitations and constraints, and there's many different things that have their limitations in terms of relative concentrations. And again, as you're pointing out here, there's a difference between simply changing hexane to octane, which is a slightly longer chain length, right? But there's an almost infinite number of surfactants, because again, surfactant being a surface active agent, so it isn't just a simplistic molecule we're talking about, it's anything that has that effect, which could be
proteins, themselves infinitely complex. And so I guess the point of all this is the ability for, at any point, to be able to accurately measure whatever system we're talking about is extremely limited if we're talking about a biological system. But the number of things that could be at play here as well from a precision diagnostic perspective, there's no way we're ever going to know what the state of play is at a particular point in time. But I guess we can look at the outcome of this and say, well, look, if the glycocalyx is forming and managing to sort of work to a degree, then the resulting systems that help create the environment, the body itself, if you like, has found a way to sort of get to that point.
You can change the nanostructure. You know, if you understand the forces operating, you can change them. Like if you change from bromide to chloride, or if with the glycocalyx, if you change from sodium to potassium, which is really interesting, you've changed the structure completely. But you know how to change them. Within the class of ions, you know how to vary them from lithium, sodium, potassium, rubidium, quaternium, ammonium. You know with lipids, you change the surfactants, the hydrocarbon region, you change the chain length, and you can vary the properties and vary them enormously, prodigiously. So you can tune them up, and you can tune them up with things called drugs that go into the surface and change.
Well, let's not jump straight into that just yet, but I guess that's what I'm alluding to in the terms of, like, there's a degree of complexity in there that we don't necessarily need to know and understand from a precision measurement of all the components into it, because going back to what you were saying just a second ago is, if you understand the basic principles of what things make what changes, then you can start to look at interventions that are more likely to...
So, that's interesting, because, see, with the microemulsions, people thought until about the 1980s that it would take 200 years to work out the nanostructure. They had armies of poor Indian students that they imported into countries like the USA, making phase diagrams, mixing this one, that one.
I see.
And that was driven in that time with the need to do fracking, because people thought we were running out of oil. So they made all these different diagrams, taking millions and millions of flasks and filling them up and making these diagrams. But there was no systematics. They couldn't, so they were searching a bit. But then people came along, me and other people, and worked out a simple way of doing it, to cut through it. They were working with 5-dimensional things. And it all turns into something really very simple, and it's organised, and you can do it. The same with the Teflon, with the polymers. It's only very recently, unfortunately for biologists and biochemists, that physical chemists have been able to get their stuff together and to draw up a unified picture of what's going on, a simple unified picture. And then it opens up big vistas.
Okay, so again, the reason for going through those on the surface of relatively complex sort of looking diagram is really to sort of highlight that point of saying everything can be going along apparently fine within those areas of concentration until you hit a sort of threshold, and then those changes can be suddenly and significantly or very dramatic, right, in terms of the effect of that. And you can measure those effects because you can measure viscosity, you can measure conductivity, etc. And we know the nature of what those things are because we know how those structures form from those self-assembly principles. So we're not just going, oh wow, suddenly there's this increase in conductivity. We know why there is because we know how those channels and everything.
Yeah, we can change those forces, and you're right. The Teflon and the microemulsions are interesting in that they're random by continuous geometries. The others we were talking about, the cubic phases formed by lipids, they're organised. They're crystalline, if you like. They're liquid crystals. But the bulk of these organelles and so on are random connected conduits and so on. So, and you're not aware of that because at least with surfactants that we talk with cubic phases, and with proteins, which are polymers, you can do X-ray crystals, crystallography.
Yes.
So what looks like an enormously complicated mess, you can pin down the structure if you crystallise them.
Yes.
In fact, they're not really crystalline, they're alive and well, but random nature of these structures makes it very difficult to explore them with simple tools, well, tools like neutron scattering.
Yeah, so just to clarify what you're saying there, I think, is that because you can sort of, in a sense, with those crystalline structures, you can take a series of snapshots, photographs, for want of a better term, that you can then analyse to say, oh, look, we're getting this consistent degree of structure forming as a result of these relatively simplistic scenarios. But when we move into the more complicated random, it's random in the sense that there's so much complexity in there and so many things that can drive it, is we either don't get a consistent outcome in the sense of something that we can take snapshots of.
Well, you can't take the snapshots, but the structure is there. It's difficult to analyse a random structure.
You can show, and I think that's what we're trying to do here, is demonstrate why the structure is there and why the measurement something makes sense.
The structure is determined by simple principles.
Yes. Yeah.
Surface.
Themselves can be demonstrated consistently.
So the whole area that we're talking about, the fuzzy layer of cells, the endothelial surface layer and glycocalyx, which is so big and so important, it's been ignored because it's just a fuzzy layer. You couldn't find the structure. And it's only by looking at analogous things like Teflon that you can say, ha-ha, this is what happens. It fits together with surfactant principles of assembly. And it's all quite simple in terms of what's driving the thing. Surface forces versus geometric packing. And that's enough.
Yeah, and so that speculation, if you like, of the glycocalyx structure is as demonstrated as possible by using all of these principles themselves that can be demonstrated, and using Nafion as sort of saying, well, hang on a minute, here's something that not only is it behaving exactly as we would expect based on this analogy, but we can demonstrate how and why it's behaving that way because we have those underlying principles that you're talking about.
I'll just say, by the way, the very fine work that people have done for many years on X-ray structure of proteins, it's very misleading in one way because they crystallise the thing and so they get a snapshot of the structure when it's crystalline. But in vivo, in the real system, they're alive and well, and they're breathing and they're expanding and so on. They're forming these random structures that have got rhyme and reason to them, but you look at the crystal structure and you think of them as rigid. They're not. They're dynamic, like the glycocalyx.
Yeah. So the way I like to think of that is it's almost like either a bunch of photos or the strobe effect. You're watching a dance before you're watching Swan Lake with a strobe light, and if Swan Lake goes for this specific period of time, and you always take this measurement, everyone's always in this position, right? And you think, oh, okay, well I noticed that's the position where the audience applauded, right? So that structure is when the audience applauds, and you're like, well, no, if you understand that it's a performance going on over time, whatever, and that's just something that when that structure forms, that's when, going back to when we were talking about your cavitation and your enzymatic reaction scenario, is there's a whole bunch of dances that could be occurring that we have no way of seeing because we can't see the whole dance. All we can see is slices, and they're very accurate slices now when they're seen, but they are still slices of an otherwise, we're taking a snapshot of a movie, if you like.
Yeah, and the movie's always the same.
Yeah. It's always Swan Lake, right? So yeah, it always goes for the same length of time. It's always got the same people and the same characters, and so we give this impression of consistency as opposed to because we haven't stepped back and say, well, hang on a minute, would it stay in that crystalline structure in that environment consistently? And what you're saying is no, because the environment itself is so dynamic. Yeah. So that's a really important, again, step back and to have in your mind when you're thinking about these things. I want to just talk a little bit about now the effect, particularly of the specific ion effects in relation to the swelling of Nafion and the effect of deuterium. In fact, maybe even if we start with deuterium, because one of the really interesting effects of this was looking at changes of deuterium levels in again, in the aqueous solution, if you like, and the effect of that on the chirality and therefore ultimately the exclusion zones in Nafion. So again, deuterium being heavy water or D2O, or water with a hydrogen with a neutron, so twice the mass, twice the size essentially of a normal hydrogen ion, and that has very different properties, very different effects when it comes to particularly the interaction with the ortho and paramagnetic versions of normal water. Do you want to just sort of walk people through the deuterium, what it is, how it affects and how it changes, and the resulting effect on things like chirality and the exclusion zone?
Well, first of all, chirality means the molecule can have two forms, left-handed and right-handed.
Sort of, so with these sorry, just to clarify which way.
Not necessarily helical, just
sure.
the original experiments of Pasteur of the 1880s, where he looked at, what did he look at? Anyway, crystallised this molecule, and they form a right-handed and left-handed form. And the right-handed and left-handed form are mirror images of each other. Mm-hmm. So the question is why they form these different kinds, because the left-handed molecules are the ones that we use in biology.
Yes.
And the right-handed ones most often will kill you. So they're just mirror images.
So just to clarify, we're talking about here specifically in terms of proteins, right? So L-tyrosine or in terms of the structure of the protein, if you took a right-handed tyrosine, it's just a mirror image of the left-handed tyrosine, but it's,
And those differences are very slight. For example, mannitol is a sugar or an alcohol sugar, and sorbitol is its mirror image, but the difference is only in one hydroxyl in hundreds of hydroxyls in the position of one of them. Yeah. It's hardly any difference, so why the hell would they be so different? Sorbitol is mostly prune juice, and it'll give you the...
Help clear you out. Yeah. Mannitol won't.
Well, it does a bit, but it's very complicated, and it affects things like Parkinson's disease very dramatically, which form you have. So it's a fundamental problem of life itself, and so why does it occur? And nobody knows, still don't know properly, but it seems that it's due to something that hasn't been given much notice until fairly recently. It seems to be due to deuterium. Deuterium is heavy water. It's hydrogen's got two. It's instead of being H2O, it's D2O. With deuterium is a proton plus a neutron instead of just a proton. So it occurs naturally in nature at quite a large quantity. It's about 160 parts per million, which is it's about 2 times 10 to the minus 2 molar, which is quite a lot, because the amount of sodium in the blood is 0.15 moles per litre. A mole is the molecular weight in grams, and deuterium is 0.2. So it's about the 10th of the concentration of sodium in the blood. Yeah. People always ignore it. Yeah. And it's like the fuzzy layer, they ignore it, okay. Yeah. But it's there and it's doing things.
Well, it's very easy to ignore until you do that math, right?
Yeah, well,
until you realise that you are almost all water,
yeah.
And a reasonable, therefore you don't need much of your water to be deuterium, and you're going to have a lot of deuterium in you.
Now there's a developing field, and I'm familiar with it with several people I'm working with now. Stephanie Senoff and Laszlo Boris have been voices crying in the wilderness. There's a whole lot of people now, a lot of Russians too, recognising this is a...
Well down the deuterium rabbit hole.
Yes. Well, it's not a rabbit hole, it's a hidden
variable. Yeah. Well, I mean that in the sense that there's a hell of a lot to it.
There's a whole lot to learn.
Yeah.
It's a new element in the whole thing. And it seems to be connected to chirality and determined chirality. Yeah. So we're hoping to get more experiments done on that. It's as you said, naturally occurring water, or water's got two forms, ortho and para. The ratio about 3:1 in gas form. And there's paramagnetic and orthomagnetic. And they compete for hydration with water lining up around ions, sulfate, chloride and so on in different ways. So the two forms compete differently, and deuterium knocks them out, and it deuterates different ions, and that determines the nature of bonds between them. So with Nafion forming a polymer, deuterium comes along, it displaces the ortho and the para water, and that changes the binding of an ion like potassium to sulfate in the helical Nafion structure, different for potassium and sodium and lithium, dramatically different.
Going back to the effect we were just sort of looking at, yeah.
So it looks as though, and this stuff started up with a fellow called Josef Skolnick some years ago, a very nice paper which more or less proves that chirality can be traced to deuterium. So that has all kinds of implications medically as well, because some bacteria kick off deuterium.
It seems to be this sort of almost biological sorting arrangement of, in particularly in the microbiome, right?
And in mitochondria too. So it's a whole new area that I'm not familiar with, but my colleagues are, and it's developing fast, and it's implicated in cancer as well. And it shows up in the Nafion case quite dramatically.
Yeah, so just to sort of backtrack a little bit and clarify. So again, what we're talking about here is we're talking about particularly these sulfonated moieties, right? So we've got these head groups that are really hydrophilic, okay? And what that means is they're essentially trying to get as close and as, associated with the water molecules as possible. But those water molecules themselves are ortho and paramagnetic. But then you've got this deuterium that sort of comes along as well that complicates the picture. And then you've also have these specific ions. We'll talk about, say, sodium as an example. So sodium is also now going to have an affinity with these moieties. So you've got all of these different things competing, and depending on the relative concentrations, you're going to get a different effect of how they all relate or
yeah, there's a slight difference in each hydration of each moiety in Teflon. In Nafion, and the combined effect, a small change to the pitch of the helix will make it wind up or down. Yeah. And the inwardness of this work we did on that was because the Nafion exclusion zone is so large, you can probe it and you can see big effects, whereas you can't see that with mitochondria or metabolism going on in the cell. You just see the effects, that's all.
Yeah, well, you can only see them when they're dead, so they're not. Yeah.
No, you can't even see them when they're dead. But you can, they're operating because the same things happen.
Because of those basic principles.
Yeah, but we know that it's operating too with heparin sulfate and the glycocalyx polymers. It's the same as Teflon. And so that changes the nature of depending on whether you've got naturally occurring water with deuterium or not, take away the deuterium. Then you get dramatic effects on the glycocalyx, and you get dramatic effects on metabolism, and you get dramatic effects on cancer too.
So let me just, before we jump back to that, I want to sort of just walk through now just to explain in a bit more detail exactly what we're referring to here and the relative effect. And there's two bits to this particular paper that I think are the key bits. It's quite a complex paper in many respects, but there's two things in here that I think are really, really important to get your head around. And one is this table here, which is looking at specific ion concentrations and the relative effect of that. And again, what needs to be appreciated from here is we're looking at levels of deuterium in water and deuterium depleted water, which is our DDW, and the effect of deuterium and specific ions and the effect of swelling. I guess that's as deep as we need to sort of go on that. And I guess the point of this is that deuterium has an effect universally in the sense that if there's no deuterium versus, let's say normal deuterium versus relatively higher levels of deuterium has an effect on its own. But then changing the ions with the same level of deuterium has an effect as well. Is that?
Yeah, at a given amount of deuterium, you get a different effect, a different level of effect between the different specific ion effects.
Yeah, and here we're measuring the exclusion zone, right? So the effect of the...
Yeah, it shows that we're using the exclusion zone to show up the effect.
Yes.
And it's highly significant. It's an unexplored area, really, because the differences between potassium and sodium are very large. They show up dramatically with the exclusion zone with Nafion. And they'll do the same with the endothelial surface layer.
Yeah, so again here, for sodium versus potassium chloride, we're talking...
It's a factor 2, but it shows up in a dramatic, in the swelling of the exclusion zone. And it does the same we've done the same for heparin and chondroitin sulfate, which are very, very analogous to the Teflon. But that's the inwardness of that is that inside cells, the salt is always potassium as the dominant cation, 98%. Outside, it's sodium. Mm-hmm. In the bloodstream, it's sodium. Inside cells, it's potassium. So the differences between water, which is normal water, which has got 2 times 10 to the minus 2 molar deuterium, and deuterium-depleted water, you can take the deuterium out, are quite dramatic in terms of the structure of the...
In fact, you can even collapse the exclusion zone.
Collapse it, yeah.
Just by changing the levels of deuterium.
Yeah. In the same way that you can collapse the deuterium layer by changing the amount of sodium and potassium, which is...
Because that's essentially what this exclusion zone is showing, is that the greater the exclusion zone, the bigger the effect, and that can go essentially to collapsing by changing the specific ion concentrations and or changing level of deuterium, yeah? Yeah, and again, what we're talking about is if we are talking about this dynamic structure of channels that flow ions, etc., and they then collapse, you can imagine, you know, the whole traffic system's been shut down.
That's right.
Yeah, and that can either be done, going back to my sort of swinging doors analogy, that can either be done temporarily, and again, we'll look at this when we go back to looking at some of those, that work particularly that Hans Vink's done in sort of showing the collapsing of the glycocalyx and the potential effect of that temporarily, through to eventually, if your glycocalyx is sort of permanently impaired in terms of we're getting through to things like atherosclerosis, where we're getting calcification of that glycocalyx region, the hardening of the arteries and those sorts of things, that now that all this functioning isn't going to occur now at anything like normal and is potentially irreversible if you have that almost sort of permanent destruction of this fuzzy layer, if you like.
Yeah.
So the other thing with this particular paper that I really like is the picture. What I refer to the bubble. Yeah. So this particular experiment here. So just make that a bit smaller. So so this is I find this a fascinating effect of the exclusion zone property, because what Nikolai was able to show here, was the effect of just simply deuterium-depleted water versus ordinary water, and the forming of essentially a bubble, right, as a result of that. And the simplistic effect of this is, with deuterium-depleted water, there is no bubble, but but in ordinary water, these bubbles kind of form, and just changing the deuterium, showing the effect of that exclusion zone sort of like barrier, if you like, forming with the level of deuterium.
Yeah, it swells differently depending on deuterium content, in the same way that it swells differently depending on potassium, sodium. They all are dependent on each other.
I guess the thing I like about this is there's one thing to use sophisticated, instrumentation and measuring equipment to sort of, with various light scattering and all those things to sort of show those effects even at that kind of level. But when a bubble pops out of nowhere just by changing the level of deuterium, then that's a pretty significant sort of, it's similar to cavitation forming in solutions. It's like
microemulsion forming.
Something significant is obviously occurring. And going back to your points around those, that a threshold has been flipped, And again, it was fine to a point, and then all of a sudden we get this huge physical difference in terms of that.
Manifestation, a different phase, but it doesn't require a lot of energy.
Not if it's at that balancing point you talk, as you say, you can flip backwards and forwards between those, as long as that sort of, homeostasis, for want of a better term, as long as that's there, the ability to flip these seemingly very substantial and very significant, completely restructuring the road network on and off in its entirety is done with virtually no energy backwards and forwards because of the balancing of these structures, and therefore maintaining those balances or how we interfere with those balances can matter. So, okay, so I think that's kind of where I wanted to get to at this sort of stage, because I think hopefully now we've laid the foundations to understand not just what the glycocalyx is in terms of this sort of dynamic structure and the forming of these channels, etc., but also how many of these different things can affect that. Because I think in the next video we want to start looking at then some examples of how that plays out, and particularly looking at things like drugs and the effect of certain types of medical interventions, right through to things looking at things like insulin sensitivity and some of those. Because if we understand this structure, those things I think start to fall into place reasonably simplistically, at least in my mind, and you can start to see how many of these phenomena that are not very well explained or not explained at all are relatively easy to comprehend and understand in relation to changes in the structure itself and how those manipulations might occur. So that's what
that's the idea, that the nature of the forces between the different molecules, very complicated molecules, that's all pretty well understood at one level. But in isolation, knowledge of those forces and knowledge of those enzymes and so on, it's a bit hampered if you don't know the infrastructure that's in the background. And that's what we're trying to do. We're elucidating the infrastructure and showing that there is a systematics to it. It's not simple at all. However, once you understand the principles that are involved, they're not very difficult then at all. Makes sense, and it's got some predictability about it.
Yeah, I guess that's a really important point, because that's, I guess, what we'll look at next is we should therefore have some predictability around interventions of certain sort of chemicals or interventions will have certain effects, and how does that actually play out based on this model. So that's, we'll really get into some really interesting stuff then, even down to things like acupuncture and hibernation, some of those really interesting things. So, all right, so we might leave that there for now, but is there anything else you want to say about?
No, I probably said too much.
I doubt anyone watching now has got anywhere near enough information if they've lasted this long. So, all right, so until next time, we'll leave it there.
Okay.